Hello AP Biology students, let's go over the 7th section in Unit 6. And yet again, I'm adding a section here for Unit 6.7. I wanted to go over a little bit more details about how proteins fold. I go over a lot of protein folding when we cover it in Unit 1 with protein structure, but I thought this would be a great chance just kind of review how proteins fold and just what the main goal of the central dogma is. Remember, there are 4 different levels of protein structure. You have primary, secondary, tertiary, and quaternary. The primary structure is just the amino acid sequence that we just talked about. Remember that this amino acid sequence is going to be determined by the DNA. Yes, the DNA is going to be copied into mRNA, which is later read for the amino acid sequence, but it's the original DNA that's going to be the instructions for a protein. So again, primary structure is just the amino acid sequence, just like the letters in a sentence. The secondary structure is going to be when those amino acids are going to start interacting with each other and forming things called hydrogen bonds. These hydrogen bonds will either create something called an alpha helix or a beta pleated sheet. These are just going to be the 2 structures that we identify as being the secondary level of a protein. The tertiary structure of a protein is when these alpha helixes and beta pleated sheets start folding on each other and start interacting to form a globular protein. This globular structure is going to have some structure to it, which is going to determine its function. Now, some proteins end here at the tertiary level. They only have one polypeptide chain. However, we have something called quaternary level. This is where some proteins are going to have several polypeptides. They're going to come together to form a larger protein. But remember, not all proteins have a quaternary level. It's just when there are several polypeptides, that is when we constitute as having a quaternary level. Just like I said before, remember that the DNA is going to code for the amino acid sequence. And how the amino acids bond together or interact is going to be based on that sequence of DNA. Here's a better image of what those hydrogen bonds look like between the beta pleated sheets here down the bottom and the alpha helixes on the right hand side here. You can see that there's going to be interactions between the elements in each one of these amino acids that form these beta pleated sheets. Now, when we look at a protein, we typically show these beta pleated sheets as being an arrow, while these alpha helixes as being like this corkscrew. So that is one easy way to identify the secondary level, is if you see arrows and corkscrews. Something else that will help determine the structure of a protein is the environment that that protein is in. Typically, proteins are in an aqueous or watery solution like the cytoplasm, which is going to be a polar environment. So what happens is the non-polar amino acids here in red are going to fold on the inside since they want to be away from water, while the polar amino acids here in blue are going to be found on the outside since they are attracted to the water. This helps the protein fold further, having some structure again, which determines function. So just as a wrap up, DNA is going to code for the amino acid sequence of a protein. These amino acids are going to interact with each other as well as in the environment to have some structure. That structure is going to determine the function of that protein.